Benefits of broccoli is one example of metabolic benefits of certain foods. Modified nucleosides are produced by methylation and oxidative damage to the DNA. This is related to developing cancer, and that is one reason I am interested in the anti-methylation detoxifying benefits of broccoli. Modified nucleosides are not reused by cells and ultimately are excreted by the body. Increased urinary levels of modified nucleosides have been detected in numerous cancer studies. Increased levels of urinary sarcosine has been observed in prostate cancer patients and were shown to be increased during prostate cancer progression and proliferation. The prostate cancer study also showed that introduction of sarcosine induced a malignant phenotype. Fatty acid synthesis has been known to be increased in tumors, and lipids are involved in many tumor activities of all kinds. Choline, phosphocholine, phosphatidylcholine, and glycerophosphocholine are needed for cell wall synthesis and are increased in brain, breast, prostate and liver cancers. Lysophosphatidylcholines are lipid intermediates that can use to form PCs or other products of phospholipases acting on PCs. Decreased blood levels of lysoPCs have been observed in lung and liver cancer, while lysophosphatic acid has been reported as increased in ovarian cancer. Tissue levels of phosphoinositides have been increased in several tumors, and 3 phosphatidyllinositols in plasma were found to be reduced in pancreatic cancer. There are thousands of these metabolic activities going on with cancer. The analyses of lipids have shown that they play a significant role in tumor metabolism, growth, and signaling, and are an important part of cancer studies about the role of fats, both good and bad. Reference is a review of applications of metabolomics in cancer by Biol, Metabolites, September 2013, 3, 552-574. Published online July 5, 2013. Phenometabolomics: the link between genotypes and phenotypes, plant moles. Biol, 2002 48 to 155 171. Clayton T.A., Linden J.C., Cloarco, Anti-H, Charul C., Hanton G., Provost J.P., Lynette J.L., Baker J.D., Wally R.J., et al. Pharmacometabonomic phenotyping and personalized drug treatment. Nature, 2006 440 to 1073 to 1077. Holmes E., Wilson I.D., Nicholson J.K. Metabolic phenotyping in health and disease, Cell, 2008 134 to 714 717. Griffin J.L., Shocker J.P. Metabolic Profiles of Cancer Cells, Nat. Reverend Cancer. 2004 4 to 551 561. Kim Y.S., Maravada P., Milner J.A. Metabolomics in Biomarker Discovery, Future Uses for Cancer Prevention, Future Onco. 2008 4 93 102. Spratlan J.L., Circavo N.J., Eckhart S.G. Clinical Applications of Metabolomics in Oncology, a Review, Clin. Cancer.im. 2009 15 to 431 440. Fanti W., Lane A.N., Higashi R.M. The Promise of Metabolomics in Cancer Molecular Therapeutics, Ca. Appin. Moles. The. 2004 6 to 584 592. Chung Y.L., Griffiths J.I. Using metabolomics to monitor anti-cancer drugs, Ernst Scaring found. Simp. Proc. 2007 4 5578. Van der Heyden MG targeting cancer metabolism, a therapeutic window opens. Nat. Reverend Drug Discof. 2011 10 to 671 684. Kajuradu Kar. Crystal B.S. Weinschilbu M Metabolomics, a global biochemical approach to drug response and disease, Anu. Reverend Pharmacol. Toxiol. 2008 48 to 653 683. doi 10.10.146/anrf.pharm.48.1130006.094715. Warburgo on the origin of cancer cells, Science. 1956 123 to 309 314. Gayton B.R.A., Gillies R.J. Why do cancers have high aerobic glycolysis? Nat. Reverend Cancer. 2004 4 to 891 899. doi 10.1038/NRC 1478. Van der Heyden M.G., Cantley L.C., Thompson C.B. Understanding the Warburg Effect, the Metabolic Requirements of Cell Proliferation, Science. 2009 324 to 1029 to 1033. doi 10.1026/science.11608009. Vision P. Sanchez Tano S. Alcaraz Vision G. Solar M. Mesquer R. Pujol MD. Lee W.N.P. Cascanti-M. Characterization of the metabolic changes underlying growth factor angiogenic activation, identification of new potential therapeutic targets. Carcinogenesis. 2009 30 to 946 952. doi 10.1093/carcin/BGP083. Israel M. Schwartzel. The metabolic advantage of tumor cells, moles, cancer. 2011 10:70. doi 10.11861476 4598 1070. Well G.A.M. Giric FR hypoxia-induced metabolic shifts in cancer cells, moving beyond the Warburg effect, int. J. Biochem. Cell Biol. 2011 43 to 981 989. doi 10.1016/j.biocell.2010.08.009.